Multilocus sequence typing. Multilocus sequence typing (MLST) is a technique in molecular biology for the typing of multiple loci using DNA sequences of internal fragments of multiple housekeeping genes to characterize isolates of microbial species. The first MLST scheme to be developed was for Neisseria meningitidis, the causative agent of meningococcal meningitis and septicaemia. Since its introduction for the research of evolutionary history, MLST has been used not only for human pathogens, but also for plant pathogens. Principle: MLST directly measures the DNA sequence variations in a set of housekeeping genes and characterizes strains by their unique allelic profiles. The principle of MLST is simple. The technique involves PCR amplification followed by DNA sequencing. Nucleotide differences between strains can be checked at a variable number of genes depending on the degree of discrimination desired. The workflow of MLST involves 1. Data collection, 2. Data analysis, and 3. Multilocus sequence analysis. In the data collection step, definitive identification of variation is obtained by nucleotide sequence determination of gene fragments. In the data analysis step, all unique sequences are assigned allele numbers and combined into an allelic profile and assigned a sequence type ST. If new alleles and ST are found, they are stored in the database after verification. In the final analysis step of MLST, the relatedness of isolates are made by comparing allelic profiles. Researchers do epidemiological and phylogenetical studies by comparing ST of different clonal complexes. A huge set of data is produced during the sequencing and identification process, so bioinformatic techniques are used to arrange, manage, analyze, and merge all of the biological data. To strike the balance between the acceptable identification power, time, and cost for the strain typing, about 7 to 8 housekeeping genes are commonly used in the laboratories. Quoting Staphylococcus aureus as an example, 7 housekeeping genes are used in MLST typing. These genes include carbamate kinase, ARC-C, shikimate dehydrogenase, ARO-E, glycerol kinase, GLPF, guanylate kinase, GMK, phosphate acetyltransferase, TA, triose phosphate isomerase, TPI, and acetyl coenzyme acetyltransferase, ICI-L, as specified by the MLST website. However, it is not uncommon for up to 10 housekeeping genes to be used. For Vibrio vulnificus, the housekeeping genes used are glucose-6-phosphate isomerase GLP, DNA gyrase, subunit BGB, malate lactate dehydrogenase MDH, methionine tRNA synthetase METG, phosphoribacillaminomidazole synthetase per 309 dehydrogenase DTD, S-diaminopymalate. Thus, both the number and type of housekeeping genes interrogated by MLST may differ from species to species. For each of these housekeeping genes, the different sequences are assigned as alleles, and the alleles at the loci provide an allelic profile. A series of profiles can then be the identification marker for strain typing. Sequences that differ at even a single nucleotide are assigned as different alleles, and no weighting is given to take into account the number of nucleotide differences between alleles, as we cannot distinguish whether differences at multiple nucleotide sites are a result of multiple point mutations or a single recombinational exchange. A large number of potential alleles at each of the loci provides the ability to distinguish billions of different allelic profiles, and a strain with the most common allele at each locus would only be expected to occur by chance approximately once in 10,000 isolates. Despite MLST providing high discriminatory power, the accumulation of nucleotide changes in housekeeping genes is a relatively slow process, and the allelic profile of a bacterial isolate is sufficiently stable over time for the method to be ideal for global epidemiology. The relatedness of isolates is displayed as a dendrogram constructed using the matrix of pairwise differences between their allelic profiles, a burst or a minimum spanning tree MST. The dendrogram is only a convenient way of displaying those isolates that have identical or very similar allelic profiles that can be assumed to be derived from a common ancestor, The relationships between isolates that differ at more than 3 out of 7 loci are likely to be unreliable and should not be taken to infer their phylogeny. The MST connects all samples in such a way that the sum distance of all branches of the tree is minimal. Alternatively, the relatedness of isolates can also be analyzed with multilocus sequence analysis (MLST). This does not use the assigned alleles, but instead concatenates the sequences of the gene fragments of the housekeeping genes and uses this concatenated sequence to determine phylogenetic relationships. In contrast to MLST, this analysis does assign a higher similarity between sequences differing only a single nucleotide and a lower similarity between sequences with multiple nucleotide differences. As a result, this analysis is more suitable for organisms with a clonal evolution and less suitable for organisms in which recombinational events occur very often. It can also be used to determine phylogenetic relationships between closely related species. The terms MLST and MLSA are very often considered interchangeable. This is, however, not correct, as each analysis method has its distinctive features and uses. Care should be taken to use the correct term. Comparison with other techniques. Earlier serological typing approaches had been established for differentiating bacterial isolates, but immunological typing has drawbacks such as reliance on few antigenic loci and unpredictable reactivities of antibodies with different antigenic variants. Several molecular typing schemes have been proposed to determine the relatedness of pathogens such as pulsed field gel electrophoresis (PFG), ribotyping, and PCR-based fingerprinting. But these DNA banding-based subtyping methods do not provide meaningful evolutionary analyses. Despite PFG being considered by many researchers as the gold standard, many strains are not typable by this technique due to the degradation of the DNA during the process gel smears. The approach of MLST is distinct from multilocus enzyme electrophoresis (MLE), which is based on different electrophoretic mobilities (EM) of multiple core metabolic enzymes. The alleles at each locus define the EM of their products, as different amino acid sequences between enzymes result in different mobilities and distinct bands when run on a gel. The relatedness of isolates can then be visualized with a dendrogram generated from the matrix of pairwise differences between the electrophoretic types. This method has a lower resolution than MLST for several reasons, all arising from the fact that enzymatic phenotype diversity is merely a proxy for DNA sequence diversity. First, enzymes may have different amino acid sequences without having sufficiently different EM to give distinct bands. Second, silent mutations may alter the DNA sequence of a gene without altering the encoded amino acids. Thirdly, the phenotype of the enzyme can easily be altered in response to environmental conditions and badly affect the reproducibility of MLE results. Common modifications of enzymes are phosphorylation, cofactor binding, and cleavage of transport sequences. This also limits comparability of MLE data obtained by different laboratories, whereas MLST provides portable and comparable DNA sequence data and has great potential for automation and standardization. MLST should not be confused with DNA barcoding. The latter is a taxonomic method that uses short genetic markers to recognize particular species of eukaryotes. It is based on the fact that mitochondrial DNA, mtDNA, or some parts of the ribosomal DNA cistron have relatively fast mutation rates, which give significant variation in sequences between species. mtDNA methods are only possible in eukaryotes as prokaryotes lack mitochondria, whereas MLST, although initially developed for prokaryotes, is now finding application in eukaryotes and in principle could be applied to any kingdom. Advantages and applications. MLST is highly unambiguous and portable. Materials required for ST determination can be exchanged between laboratories. Primer sequences and protocols can be accessed electronically. It is reproducible and scalable. MLST is automated, combines advances in high-throughput sequencing and bioinformatics with established population genetics techniques. MLST data can be used to investigate evolutionary relationships among bacteria. MLST provides good discriminatory power to differentiate isolates. The application of MLST is huge and provides a resource for the scientific, public health, and veterinary communities as well as the food industry. The following are examples of MLST applications. Campylobacter. Campylobacter is the common causative agent for bacterial infectious intestinal diseases, usually arising from undercooked poultry or unpasteurized milk. However, its epidemiology is poorly understood since outbreaks are rarely detected, so that the sources and transmission routes of outbreak are not easily traced. In addition, Campylobacter genomes are genetically diverse and unstable with frequent and intragenomic recombination together with phase variation, which complicates the interpretation of data from many typing methods. Until recently, with the application of MLST technique, Campylobacter typing has achieved a great success and added onto the MLST database. As at 1 May 2008, the Campylobacter MLST database contains 3,516 isolates and about 30 publications that use or mention MLST in research on Campylobacter http://published.org/Campylobacter/ Neisseria meningitidis. MLST has provided a more richly textured picture of bacteria within human populations and on strain variants that may be pathogenic to human, plants, and animals. MLST technique was first used by Maiden et al. 1 to characterize Neisseria meningitidis using 6 loci. The application of MLST has clearly resolved the major meningococcal lineages known to be responsible for invasive disease around the world. To improve the level of discriminatory power between the major invasive lineages, 7 loci are now being used and have been accepted by many laboratories as the method of choice for characterizing meningococcal isolates. It is a well-known fact that recombinational exchanges commonly occur in N. meningitidis, leading to rapid diversification of meningococcal clones. MLST has successfully provided a reliable method for characterization of clones within other bacterial species in which the rates of clonal diversification are generally lower. Staphylococcus aureus. S. aureus causes a number of diseases. Methicillin-resistant S. aureus MRSA has generated growing concerns over its resistance to almost all antibiotics except vancomycin. However, most serious S. aureus infections in the community, and many in hospitals, are caused by methicillin-susceptible isolates MSSA, and there have been few attempts to identify the hypervirulent MSSA clones associated with serious disease. MLST was therefore developed to provide an unambiguous method of characterizing MRSA clones and for the identification of the MSSA clones associated with serious disease. Streptococcus pyogenes. S. pyogenes causes diseases ranging from pharyngitis to life-threatening impetigo, including necrotizing fasciitis. An MLST scheme for S. pyogenes has been developed. At present, the database MLST.net contains the allelic profiles of isolates that represent the worldwide diversity of the organism and isolates from serious invasive disease. Candida albicans. C. albicans is a fungal pathogen of humans and is responsible for hospital-acquired bloodstream infections. MLST technique has used to characterize C. albicans isolates. Combination of the alleles at the different loci results in unique diploid sequence types that can be used to discriminate strains. MLST has been shown successfully applied to study the epidemiology of C. albicans in the hospital as well as the diversity of C. albicans isolates obtained from diverse ecological niches including human and animal hosts. Cronobacter. The genus Cronobacter is composed of 7 species. Before 2007, the single species name Enterobacter sakazakii was applied to these organisms. The Cronobacter MLST was initially applied to distinguish between C. sakazakii and C. melanoticus because 16S DNA sequencing is not always accurate enough and biotyping is too subjective. The Cronobacter MLST scheme uses 7 alleles at D, FUS-A, GLNS, GLTB, GB, INFB, and PPS, giving a concatenated sequence of 3036 BP for phylogenetic analysis, MLSA, and comparative genomics. MLST has also been used in the formal recognition of new Cronobacter species. The method has revealed a strong association between one genetic lineage, sequence type 4 ST4, and cases of neonatal meningitis. The Cronobacter MLST site is at http://www.pubmlst.org/Cronobacter. Limitations. MLST appears best in population genetic study, but it is expensive due to the sequence conservation in housekeeping genes. MLST sometimes lacks the discriminatory power to differentiate bacterial strains, which limits its use in epidemiological investigations. To improve the discriminatory power of MLST, a multi-virulence locus sequence typing MVLST approach has been developed using Listeria monocytogenes. MVLST broadens the benefits of MLST but targets virulence genes, which may be more polymorphic than housekeeping genes. Population genetics is not the only relevant factor in an epidemic. Virulence factors are also important in causing disease, and population genetic studies struggle to monitor these. This is because the genes involved are often highly recombining and mobile between strains in comparison with the population genetic framework. Thus, for example, in Escherichia coli, identifying strains carrying toxin genes is more important. The advent of second-generation sequencing technologies has made it possible to obtain sequence information across the entire bacterial genome at relatively modest cost and effort, and MLST can now be assigned from whole genome sequence information rather than sequencing each locus separately, as was the practice when MLST was first developed. Whole genome sequencing provides richer information. For differentiating bacterial strains, MLST uses approximately 0.1% of the genomic sequence to assign type while disregarding the rest. For example, whole genome sequencing of numerous isolates has revealed the single MLST lineage ST258 of Klebsiella pneumoniae comprises 2 distinct genetic clades, providing additional information about the evolution and spread of these multi-drug-resistant organisms and disproving the previous hypothesis of a single clonal origin for ST. Databases. MLST databases contain the reference allele sequences and sequence types for each organism and also isolate epidemiological data. The websites contain interrogation and analysis software, which allow users to query their allele sequences and sequence types. MLST is widely used as a tool for researchers and public healthcare workers. The majority of MLST databases are hosted at 2 web servers currently located at Imperial College, London MLST.net, and in Oxford University PubMed.org. The databases hosted at each site are different and hold the organism-specific reference allele sequences and lists of ST for individual organisms. To assist the gathering and formatting of the utilized sequences, a simple and free plug-in for Firefox has been developed. Link. Journal articles. Made in MC by Graves Ya, Fail et al. March 1998. Multilocus sequence typing, a portable approach to the identification of clones within populations of pathogenic microorganisms. Croc. Natal. Acad. Sky. USA 95-6, 3145. Bipode 1998 PNAS. 95.3140M. DOI 10.1073/PNAS 95.6. 3140. PMC 19708. PMID 9,500,229. Owen R. Made in MC, made in October 2003. Multilocus sequence typing, a tool for global epidemiology. Trends microbial. 1110, 47987. DOI 10.1016/J Tim 2003.8 6 PMID 14,557,031 Foley SL White DG McDermott PF et al. October 2006 Comparison of subtyping methods for differentiating Salmonella enterica-cerevar typhimurium isolates obtained from food animal sources. J. Klin, Microbial, 4410, 356977. doi 10.1128/JCM 745-6, PMC 1,502,788, PMID 17,021,084. Johnson J.K. Arduino SM Steino C. Johnson Ya. Harris A.D. Arduino Stein. Johnson. Harris November 2007. Multilocus sequence typing compared to pulsed field gel electrophoresis for molecular typing of Pseudomonas aeruginosa. J. Klin. Microbial. 4511 3707-12. doi 10.1128/JCM 560-7. PMC 2,168,506. PMID 17,881,548. CSON maint. Multiple names. Authors list link. Nalaperdi SR. DRW Singh KV. Murray B. Da Singh. Murray March 2002. Molecular typing of selected Enterococcus faecalis isolates. Pilot study using multilocus sequence typing and pulsed field gel electrophoresis. J. Klin. Microbial. 43 868-76. doi 10.1128/JCM 40.3 868-876.2002. PMC 120,268. PMID 11,880,407. CSON maint. Multiple names. Authors list link. Thacker and K. Nalan LK log CM Nalan log May 2005. Multilocus sequence typing lacks the discriminatory ability of pulsed field gel electrophoresis for typing Salmonella enterica-cerevar typhimurium. J. Klin. Microbial. 43.5 2215-9. doi 10.1128/JCM 43.5 2215-2000 219.2005. PMC 1,153,745. PMID 15,872,244. CSON maint. Multiple names. Authors list link. Chen Y. Zhang W. Mabel S.J. Zhang Mabel October 2005. Multivirulence locus sequence. Typing clarifies epidemiology of recent listeriosis outbreaks in the United States. J. Klin. Microbial. 4310 5291-4. doi 10.1128/JCM 43.10 5291-5000 294.2005. PMC 1,248,515. PMID 16,208,000. CSON maint. Multiple names. Authors list link.